Okay, hello everyone. My name is Eli. I'm a postdoc here at the University of Waikato. I'm working on a genomics Aotearoa funded project, Invasomics for Biosecurity. In this project, we aim at identifying genomic signature of invasion success at phylogenetic and population genomic levels in exemplar pest insects. Amy is the other postdoc in this project, and she's focused on the phylogenetic aspect of the project. And she will present her work in the afternoon. I'm more focused on the population genomics side. And today I will present my work on the brown marmorated stingbug. So as already mentioned during this conference, we know that invasive species can undergo adaptive evolutionary change in response to changes in biotic and abiotic conditions in their newly colonized environments. Understanding the genomic underpinnings of adaptive evolutionary change can sometimes be challenging because selective sweep tests often require high genomic marker density, reference genome contiguity, and direct estimates of genome-wide mutation and recombination rates. Despite these challenges, the increasing availability of annotated pest genomes, for example, through the I5K project, the affordability of high-throughput sequencing, such as rat sequencing, and advances in population genomic approaches together provide novel opportunities to study the genomic underpinnings of invasion success in insect pests. For example, rat sequencing data can be used in genome-wide scanning, as mentioned earlier, to characterize, to detect shifts in allele frequencies between different populations. And specifically for invasive species, the historical status of populations, whether native or invasive, can be considered as a binary covariate in genome-wide scanning to characterize association of genomic variants with invasive status of populations. This approach has recently been introduced in the program BayPass. Louise also mentioned that in her talk. And this approach contrasts allele frequencies between native and invasive populations while accounting for or correcting for shared demographic histories across populations. Functional annotation of the SNPs that are identified under this approach can provide insights into genes and physiological functions potentially associated with invasion. So the focus of my work today is the brown marmorated stingbug, or BMSB. BMSB is native to East Asia, including China, Korea, and Japan. It is a highly invasive agricultural pest. It's capable of feeding on more than 100 plants, including economically important crops. It is an opportunistic hitchhiker, and a large number of adults can aggregate in shipping containers and imported food and wood. It was first reported outside its native range in Pennsylvania in 1996, and since then it has spread into other parts of the US and also into other countries. For example, it started invading Europe since 2004. It was reported in Canada in 2008, and also in South America in 2017. Currently, it is widespread in parts of the US, Italy, Switzerland, and Slovakia, but is localized in other countries. It's also frequently intercepted at the borders in New Zealand. Previous mitochondrial DNA studies suggest that China is the most likely source of global invasion, with multiple out-of-China invasion events, and also bridgehead events from the US to Europe and within Europe. Ecological niche modeling suggests that Europe, northeastern North America, parts of South America, Africa, Southern Australia, and the North Island of New Zealand are at high risk for further colonization of BMSB. It has also been suggested that rising temperatures under future climate change scenarios can accelerate this range expansion by accelerating population growth and increasing the number of generations that BMSB can produce annually. So in this study, we aim at identifying highly differentiated SNPs or outlier SNPs by using genome-wide scanning. These differentiated SNPs are different between native and invasive populations, obviously. We are also interested in highlighting the genes that lie in close proximity of outlier SNPs and therefore may be of potential invasive interest. We also did population assignment of intercepted samples at New Zealand borders to see whether the country or geographic region that these samples have originated from can be identified using RAD sequencing data. So just a quick summary of the methods that we had. We had double digest RAD sequences of 404 BMSB samples from native, invasive, and intercepted ranges. We did population structure, admixture, and gene flow analysis. We used the program AssignPop, which uses a machine learning approach for the assignment of New Zealand intercepted samples. We used BayPass for genome-wide scanning for association with invasiveness. And finally, by using the BMSB assembly that was published in 2020, we functionally annotated outliers. So these results are based on around 14,000 hard-filtered SNPs. We found that within the native range in Asia, BMSB is highly structured. Japan and China form very two distinct clusters. But within the invasive range, the levels of admixture are high, and there's a lack of discrete population structure that can be associated to a certain country or even geographic region, except for Romania and to some extent to Georgia and Turkey. We also found that the samples within the invasive range are genetically closer to China. So again, China is the most likely source of global invasion. Patterns of contemporary gene flow over the past few generations showed an overall low level of contemporary gene flow, but there are a few exceptions. There is relatively higher gene flow towards the US and Georgia from different invaded countries and towards China from a few of the invaded countries. Cross-validation results from population assignment of New Zealand intercepted samples showed that regardless of the proportion of loci or the proportion of individuals used in the training data set, the highest assignment accuracies can be obtained when samples are driven directly from the native range, either Japan or China. So the samples, intercepted samples that were found in containers that were shipped from Japan were correctly assigned to Japan, but the ones that were found in containers coming from the US or Italy were not correctly assigned to their correct country. So we see an admixture of different colors corresponding to different countries for the US and Italy. Our outlier analysis using BayPass identified 63 outlier SNPs, and using the BMSB genome assembly, we found we could locate exons around 22 of these outliers. These exons were associated with genes with products of potential adaptive importance in invasion. For example, GST, which is important in detoxification, and sodium channel proteins that are the main target of many commonly used insecticides against BMSB. We also found significantly enriched gene ontology terms for oxidoreductase activity, odorant binding, and protein binding. So in conclusion, we found that BMSB has a complex invasion history, the levels of admixture are high, and there are possibly multiple bridgehead events. This complexity reduces the efficiency of RAD sequencing data in accurately assigning incursions to their source, unless the incursions originated directly from the native range. Also, our genome-wide scanning identified genes potentially involved in BMSB's invasion success. For example, genes that are important in detoxification and insecticide resistance. There is no record of insecticide resistance in BMSB to date, but our results suggest that resistance phenotypes may have the potential to evolve from standing variation. Future studies using more SNPs, for example, by using whole genome resequencing, having an improved genome assembly, both in terms of contiguity and annotation, and also having a denser geographic sampling, including unsampled invasive populations, for example, in Europe, or unsampled populations within the native range, especially in China, which is the main source of global invasion, can provide better insights into evolutionary mechanisms underlying invasion success in BMSB. We would like to thank Plant Health and Environment Lab at MPI for providing access to New Zealand intercepted samples, GA for funding this research, and the Invasomics Lab for their support. Thank you.
Well done. Any questions for Eli?
Yep, thanks for another great talk. There's been so many great talks here. I'm curious about the outlier analysis, a bit more about that. So you're basically predicting whether the thing is native or introduced and looking for outlier SNPs.
Yeah, comparing the two groups, native versus invasive.
Right. Yeah. And is there any kind of underlying population structure model in that?
It takes into account, the program BayPass actually takes into account.
Okay, great.
Yes, it creates a matrix of covariance matrix of population allele frequencies, and then by using that, it tries to control for population structure.
Yeah, great. Thank you.
Thanks, Eli. I had a question about whether or how optimistic you might be about being able to assign populations if you had whole genome resequence data, given how much we've heard today about how maybe differentiation can be really quite localized and restricted in genomes. Do you think more data would get you there, or it's too hard?
That's one thing that we are not sure, because then the species, I think it had a lot of bridgehead events and lots of admixture events. So whole genome resequencing, that might not provide enough resolution because of all this complexity and admixtures.